Okay, so I do apologize for that. Let's continue on. We were discussing what a tyrosine kinase enzyme actually is. Okay, so they're enzymes which are capable of phosphorylating tyrosine residues within proteins. Okay, so let's show this. So I'll draw a phosphate group once again, which is a structure we're now familiar with. So here we have a phosphorus atom double bound to an oxygen atom with two alcohol groups coming off, and then an oxygen single bound to this phosphorus atom, which has then acquired an electron via ionic means and therefore has a negative charge. Okay, right. You can then add phosphate groups onto the tyrosine residues by forming phosphoester links. Okay, so you're going to take the alcohol group off the phosphate molecule, you're going to take the hydrogen off the alcohol group of the tyrosine residue, you're then going to bind the oxygen of the alcohol group of the tyrosine residue to the phosphorus atom of this inorganic phosphate group here. Okay, and this link once again will be one of these phosphoester links. Now, this isn't quite true, what I've told you here. Tyrosine kinase enzymes do not just take an inorganic phosphate from the cytoplasm and bind it onto the alcohol group of a tyrosine residue. Instead, they usually get the phosphate group off an ATP molecule. Okay, so they take adenosine triphosphate, rip off its gamma phosphate, taking it down to ADP, adenosine diphosphate, and they then push that phosphate group that they've got onto the alcohol group of the tyrosine residue. Okay, but this gets the essential principle across of what tyrosine kinase enzymes actually achieve. They achieve the phosphorylation of tyrosine residues. Okay, now, normally, these tyrosine kinase domains of receptor tyrosine kinases are extremely inactive. Okay, so they are not really catalyzing the phosphorylation of tyrosine residues within proteins. Okay, however, they do have a tiny little bit of basal activity, so they can do it, they're just extremely bad at doing it. Okay, now, when you bring two receptor tyrosine kinases together like this, and I'll stress that it's the ligand binding domains which have dimerized, not the tyrosine kinase domains, they're still separated. Okay, but what happens is these two come very close together, and what's going to happen is a process known as trans-autophosphorylation, okay, which I'll put down here. Okay, sometimes just called autophosphorylation rather than trans-autophosphorylation. Okay, because it's already a massive word without the trans. Okay, I will explain why this process is called that after I've actually explained the process. So what's firstly going to happen is that the tyrosine kinase domain on this first receptor tyrosine kinase, and for this discussion it will be very helpful to have names for these two. Let's call this number 1 and this number 2. What's going to happen is that the tyrosine kinase domain of this first receptor tyrosine kinase is going to phosphorylate a tyrosine phosphorylation site on the tyrosine kinase domain of the second receptor tyrosine kinase. Okay, so each of these tyrosine kinase domains has a tyrosine residue, which is a phosphorylation site for a tyrosine kinase enzyme. Okay, so now when they're brought really close together, the basal activity is going to result in one phosphorylating the tyrosine residue on the other. Okay, so let's say that number 1 firstly phosphorylates number 2. Okay, now when you phosphorylate the tyrosine residue within these tyrosine kinase domains, it results in activation of the tyrosine kinase domain. So this tyrosine kinase domain is now suddenly going to become really active. Okay, so it's going to become much more active. It will then reciprocate for the tyrosine kinase domain of number 1. Okay, so it will phosphorylate the tyrosine on the tyrosine kinase domain of number 1. Okay, and similarly, that will now mean that the tyrosine kinase domain of the first receptor tyrosine kinase is activated. So both of these tyrosine kinase domains are now activated. Okay, and what's now going to happen is this tyrosine kinase domain on receptor tyrosine kinase number 1, which is phosphorylated here, is now going to phosphorylate tyrosine residues that are outside of the tyrosine kinase domain on number 2. Okay, so there are also tyrosine residues in the regions of the polypeptide surrounding the tyrosine kinase domain. Okay, so like so. So I'll just draw 2 of these on each receptor tyrosine kinase, one here and one here. But in reality, there can be far more of these things. Okay, the point is that there are tyrosine residues outside of the outside of the tyrosine kinase domain, but still within this cytoplasmic tail of the receptor tyrosine kinase, which can be phosphorylated by the tyrosine kinase domain. Okay, so we now have these 2 active tyrosine kinase domains, and what's going to happen is that the tyrosine kinase domain of number 1 is going to phosphorylate all of these tyrosines within the cytoplasmic tail of receptor tyrosine kinase number 2. Okay, so this one is going to phosphorylate these 2. Okay, and similarly, the tyrosine kinase domain of receptor tyrosine kinase number 2 is going to phosphorylate the tyrosines within the cytoplasmic tail of receptor tyrosine kinase number 1. Okay, like so. Right, so this process, the full process, firstly the process of them phosphorylating each other to activate them, and then the process of them phosphorylating the other tyrosines on the cytoplasmic tail of the opposite receptor tyrosine kinase. This is known as trans-autophosphorylation. So why is it called that? Well, phosphorylation because we are phosphorylating tyrosines. Okay, so that explains that portion. Auto, what does auto mean? Well, auto means self. Okay, autonomy means self-governing. So autophosphorylation means self-phosphorylation. Okay, now the reason this is called self-phosphorylation is because if you view the dimer as being one entity in itself, then the tyrosines on this dimer are getting phosphorylated. How are they getting phosphorylated? Well, they're getting phosphorylated by the dimer itself. Okay, so if you view the dimer as one entity, then it is autophosphorylation. Now, of course, we can they sort of go back on that with this trans portion here, because then we acknowledge that actually this dimer is 2 separate proteins, and it's actually receptor tyrosine kinase number 1 phosphorylating receptor tyrosine kinase number 2, and then receptor tyrosine kinase number 2 phosphorylating receptor tyrosine kinase number 1. Okay, so trans reflects that fact that they are phosphorylating the one across from them, so trans means across from. Okay, so that's why it's called trans-autophosphorylation, or you can just call it autophosphorylation if you like. Now, it is the appearance of these phosphotyrosine residues on the cytoplasmic aspect of our receptor tyrosine kinase dimer which is going to serve to recruit our type 1a PI3 kinase enzyme to the underside of the phospholipid bilayer and to activate it slightly. Okay, so the type 1a PI3 kinase enzyme is going to bind to these phosphotyrosine residues. Okay, so let's now discuss this on the next page. Okay, right, so let's firstly redraw out our receptor tyrosine kinase dimer. Okay, so I'm going to draw this nice and big so that I've got room to draw my type 1a PI3 kinase enzyme in as well. So here is a receptor tyrosine kinase. Okay, this is the ligand binding domain here. Then we've got the membrane spanning alpha helix, and then we've got our tyrosine kinase domain intracellularly here, and then the C-terminal after that. Okay, then we've got our other receptor tyrosine kinase also out here. Here is its ligand binding domain bound to the ligand binding domain of the first one, then the membrane spanning alpha helix, then the tyrosine kinase domain, like so, and then the C-terminal, like so. Okay, and then we've got our ligand bound to the ligand binding domain here. Okay, so let's colour this in once again. So we can imagine that our ligand is epidermal growth factor, if we want a specific example in mind. We can imagine our receptor tyrosine kinase is the epidermal growth factor receptor. Here are the two ligand binding domains in orange here. Okay, then we have our membrane spanning alpha helices here in red. Okay, and here are our tyrosine kinase domains intracellularly here. And remember, in the case of the epidermal growth factor receptor and all the other B family members, these are whole tyrosine kinase domains, so they're not split. Okay, we've then firstly had the trans-autophosphorylation of the two tyrosine residues within the tyrosine kinase domain itself. Okay, which has activated the receptor tyrosine kinase domains. Okay, and they've then gone off and phosphorylated the tyrosine residues everywhere else, basically, on their two cytoplasmic tails here. Okay, right. Now, remember, our type 1a PI3 kinase enzymes, they were present within the cytoplasm initially. And remember, this was one of the reasons that they weren't actually capable of doing anything, because their substrate is in the inner leaflet of the phospholipid bilayer up here. So it's useless for them to be in the cytoplasm. Okay, what is now going to happen is the NSH2 domain here and the CSH2 domain here are going to bind to these phosphorylated tyrosine residues. Okay, and by the way, the name for a phosphorylated tyrosine residue is to call it a phosphotyrosine residue. Okay, so SH2 domains, or Sark homology 2 domains, are famous. They are famous because they can bind to phosphotyrosine residues. They are a special conserved domain that you find in a huge number of different proteins which are capable of binding to phosphotyrosine residues. Okay, so this NSH2 domain and this CSH2 domain, both of them are going to bind to phosphorylated tyrosine residues or phosphotyrosine residues. Okay, right, so this is what's going to happen. The NSH2 domain of our regulatory subunit of our type 1a PI3 kinase enzyme is going to bind to a phosphotyrosine residue. And for the basis of this picture, I'm going to ignore the breakpoint cluster homology domain and the SH3 domain. Remember, those were only present in the two big ones anyway, so we can imagine we're drawing one of the short ones. So we'll have our amino terminus after this. So we'll say we've got a short regulatory domain, so either a P50 alpha, a P55 alpha, or a P55 gamma. Okay, so here is our NSH2 domain, bound to this phosphotyrosine residue here. The CSH2 domain is also going to bind to a phosphotyrosine residue. Okay, so here it is. Okay, so at the moment we're just drawing the regulatory subunit, not the catalytic subunit yet. Okay, then in between the NSH2 domain and the CSH2 domain, remember we have these two alpha helices which coil around each other like so. Okay, known as the inter-SH2 domain, and I'm just showing these alpha helices in blue here. So this is the I-SH2 domain. Then the catalytic subunit is bound here. Okay, so here's the amino terminus of the catalytic subunit. Then remember, one of the first domains you had was the adapter binding domain here. Okay, which we'll colour in blue again. Okay, and that binds to the inter-SH2 domain. Okay, then we have the RAS binding domain, which, remember, binds to RAS monomeric G proteins that are in the on state. Now, RAS monomeric G proteins are also attached to the underside of the phospholipid bilayer, and that's how RAS G proteins serve to translocate the enzyme from the cytoplasm to the intracellular aspect of the phospholipid bilayer. And then you've got the C2 domain, okay, which we'll have here in orange. Okay, then after the after the C2 domain, you then have the helical domain here, which remember forms a interaction with the NSH2 domain here. Okay, so this is the helical domain, which I'll colour in in vivid purple, and then finally, but just before the C-terminus of the catalytic subunit, you actually have the domain which is capable of catalyzing phosphorylation of PIP2 into PIP3, which is the catalytic domain. Can I squeeze catalytic in there? Evidently not. Okay, then you have the carboxylic acid terminal here. Okay, so we will colour the catalytic subunit, well, the catalytic domain of the catalytic subunit in turquoise there, and that will also be interacting with the ISH2 domain. Okay, so we have now firstly translocated our whole PI3 kinase enzyme up to the underside of the phospholipid bilayer. In addition, you'll get slight conformational changes here in the regulatory subunit, which will then change the conformation slightly of the catalytic subunit here, which will activate it to some degree. But the main thing that this does is merely translocate it so that it's now attached to the underside of the phospholipid bilayer. The catalytic subunit can now work on PIP2 molecules which are in the inner leaflet of the phospholipid bilayer. Okay, so this is a PIP2 molecule here. So let's colour in the different portions. Here are the long chain carboxylic acids in orange. Here is the glycerol molecule in green here. Here are the phosphate groups in vivid purple. 1, 2, 3. And here are, it is the inositol ring in blue there. Okay, it's now going to phosphorylate PIP2 into PIP3, and then the PIP3 will then go on to activate protein kinase B, or lead rather to the activation of protein kinase B. It's not PIP3 that activates protein kinase B itself, but indirectly it will lead to the activation of protein kinase B. Okay, right, so that now concludes our discussion of the type 1 phosphoinositide 3-Kinase enzymes.